In previous video, you already discussed about the different types of classifications by different sciences like Aristotle classified animals into two kingdoms. There are anima and enema. This means invertebrates and vertebrates. This means non-red blood animals, red blood animals. Then after Carolus von Linnaeus tried to classify biological organisms into on the basis of scientifical, there are Plantae and Animalia. Two kingdom of classification was introduced by Carolus von Linnaeus. Okay. In addition, this Carolus von Linnaeus, Ernest Haeckel classified biological organisms into three kingdoms. What are they? Plantae, Animalia, and Protista. Okay. All unicellular organisms are categorized under this Protista kingdom. Then after Copeland classified entire biological organisms into four kingdoms. What are they? Monera, Protista, Plantae, and Animalia. Four kingdom classification. Okay. Then after R.H. Whittaker classified entire biological organisms on the basis of some criteria like cell structure, body organization, nutrition, types of cell, reproduction, and phylogenetical relationship, etc. On the basis of this criteria, R.H. Whittaker classified entire biological organisms into five kingdoms. What are they? Monera, Protista, Fungi, Plantae, and Animalia. Okay. This classification is the most acceptable classification, and majority of the sciences are accepted this classification. After R.H. Whittaker classification, Carl Woese also classified entire biological organisms into six kingdoms. He was classified Monera kingdom into two sub-kingdoms. What are they? Archaebacteria and Eubacteria. Archaebacteria and Eubacteria. Okay. Then after ICBN and ICGN also classified entire biological organisms into 8 kingdoms you already know. Okay. But in this video, I want to briefly discuss about one by one kingdom of R.H. Whittaker classifications kingdoms. What are they? Monera, Protista, Fungi, Plantae, Animalia. The first kingdom is Monera. R.H. Whittaker classification. The Monera kingdom is the initial kingdom and also prokaryotic kingdom. This means all prokaryotic organisms are classified under this Monera kingdom. Okay. This kingdom was proposed by Doty and Allen. Doty and Allen. Kingdom is a prokaryotic kingdom. It comprises some organisms like bacteria and mycoplasmas and actinomycetes, blue-green algae, BGA, blue-green algae. These are also called cyanobacteria. Okay. In this way, some bacteria, mycoplasma, actinomycetes, blue-green algae are classified under this Monera kingdom. Monera kingdom species are the very microscopic. Some are unicellular like bacteria. Mycelial organisms like actinomycetes. Colonial forms like nostrac. Filamentous like oscillatoria. The cell wall is mainly made up of peptidoglycan. The peptidoglycan material is present in this cell wall of the Monera kingdom species. Kingdom organism cytoplasm does not have any membrane bounded cell organelles. Membrane bounded cell organelles like mitochondria, chloroplast, nucleus, Golgi complex. This type of membrane bounded cell organelles are absent in this. Understand? Next one, Except ribosomes. Except ribosomes, remaining cell organelles are absent in the cytoplasm of the Monera organisms. Okay. Next one. The naked circular DNA is present. It is not enveloped by any nuclear membrane. Understand? The naked circular DNA is present. It is called bacterial chromosome or genophore. Bacterial chromosome or genophore. And the Monera kingdom organisms are consist gas vacuole. Gas vacuoles are occurred, but sap vacuoles are completely absent. Sap vacuoles are absent, but gas vacuoles are present. The cytoplasm of the Monera kingdom organisms only consist ribosomes. Understand? Only ribosome cell organelles are present, remaining cell organelles are absent in this cytoplasm of the Monera kingdom organisms. And these ribosomes are 70S type in nature. 70S type. Understand? Prokaryotic organisms are consist only 70S type of ribosomes, but eukaryotic organisms consist 80S type of ribosomes. Motility organisms of Monera kingdoms consist of flagellas. Flagella that are helpful for swimming, movement, locomotion. Understand? Because by the presence of flagella there are locomotion. These flagellas are mainly made up of one of the specialized protein. That one is flagellin. Cyanobacteria, that means blue-green algae, and photoautotrophic bacteria, that means they are consist bacterial chlorophyll. Because of this, they perform photosynthesis activity, assimilation process. Okay, both photoautotrophic bacteria and the cyanobacteria, and that means blue-green algal members are perform another. In addition to photosynthesis, they are also perform biological nitrogen fixation. This means the atmospheric nitrogen captured by this bacteria and converted into ammonia. Okay, the molecular nitrogen is converted into ammonia by these cyanobacteria and photoautotrophic bacteria. Okay, because of the performing this biological antifixation, the soil fertility also increase. Understand? Asexually, these Monera kingdom organisms are reproduced through fission, budding, fragmentation, and spores formation. Remember this one? Asexually, Monera kingdom organisms are reproduced their offsprings by fission, budding, fragmentation, and spores formation. Another one, sexual reproduction does not occur in this Monera kingdom organisms. Because of this reason, garment formation is completely absent. Okay, garment formation is absent because sexual reproduction does not occur in this Monera kingdom organisms. The last one, Monera kingdom comprises of two main groups. What are they? The first one is Archaea. It is also called Archaebacteria and Eubacteria. Archaebacteria and Eubacteria. Let us discuss about first one, Archaebacteria. In the kingdom, the first group is Archaebacteria. Archaebacteria are the primitive and ancient organisms. Hence, these are also known as living fossils. Living fossils. Next one, there are live as some extreme harshy areas or harshy conditions like hollow fields. This means these are live in the salt pans. And thermoacetophiles. This means these are can live at a higher temperature up to 80 degrees centigrade. Next one, some are methanogens. This means these are live in the marshy areas. Marshy areas. The cell wall of this archaebacteria, mucopeptide is absent. Mucopeptide is absent in the cell wall. It is replaced by another material. That one is pseudomurin. Remember this one? There are consist in the cell wall pseudomurin. Because of the presence of pseudomurin, these archaebacteria can survive in some harshy conditions like halophils, thermoacetophiles and methanogens area. Understand? The cell membrane of the archaebacteria are made of branched chain lipids. By the presence of these branched chain lipids, these archaebacteria are protected from radiations. Let us discuss about some harshy habitat condition archaebacteria. Some archaebacteria are methanogens. These are the obligated anaerobic bacteria. Obligated anaerobic bacteria. There are the organic components are converted into carbon dioxide and methane. Methane gas is helpful for the gobar gas preparation. It is helpful for cooking. It is used, highly used in the village and urbanic areas, used as cooking. Next one, some are like as symbionts. Some live as symbionts in cattle ruminant. Cattle ruminant. Example, methanogen bacteria. Methanogen bacteria. These methanogen bacteria are live in the cattle ruminant. They are utilized for the digestion of cellulose and the cattle. That means herbivorous animals. Okay. Halophils. Halophils means nothing but they are live in this rich salty areas. Salty areas. Next one. Carotenoids are present in the cell membrane. By the presence of a cell membrane, protect from the radiations also. Okay. Example, halobacteria and halococcus. Halobacteria and halococcus bacteria are example to the halophils. Thermoacetophiles. They are ability to high temperature and high acidity. Okay. High temperature till 80 degrees centigrade and acidity pH 2 value. 2 value there are can survive at the high temperature and high acidity. These thermoacetophiles are extensively found in the hot sulfur springs. Hot sulfur springs. Okay. Some example, thermoplasma bacteria. Another second group in the Monera kingdom, that one is eubacteria. Eubacteria. Okay, these eubacteria are most abundant microorganisms. Okay. Next one, there are also living some extreme habitats like archaebacteria. Next, many of these eubacteria live as parasitic organisms, causes different types of diseases both in plants and animals. Okay, so these are majority of the eubacteria live as parasites. The next one, these bacteria are first time identified by the scientist Anton van Leeuwenhoek. Okay. And the sewage water. He was prepared one microscope in two concave and convex mirrors. Under this microscope, he was observed sewage water. Okay, and our mouth wastage material. Okay, these are all sewage water. Different types of sewage waters are observed under his own microscope by the Anton van Leeuwenhoek. He was observed a small, tinny like animals. He was called them animalcules. After these animalcules are named bacteria by the Ehrenberg. This means Ehrenberg called these animalcules as bacteria. This means bacteria term is introduced by Ehrenberg. Understand? Next one. The basis of the morphological shape of bacteria, they are categorized into 4 groups. What are they? First one is coccus. These are spherical bacteria. Round-shaped bacteria are called coccus bacteria. Bacillus, these are the rod-shaped bacteria are called bacillus. Vibrio, this means half helical bacteria are called Vibrio. Next one, complete helical bacteria are called Spirillum. In this way, on the basis of their shape, bacteria are categorized into coccus, bacillus, vibrio, and spirillum. First one, round-shaped bacteria are called coccus bacteria. Rod-shaped bacteria are called bacillus bacteria. And off-helical or comma-shaped bacteria are called vibrio. And complete helical or spherical bacteria are called spirillum. These are the different types of bacteria are on the basis of their shape. Bacteria are the round-shaped spherical bacteria. These are non-motile bacteria. This means flagellae are absent in this cocci bacteria. Any cocci bacteria. Okay. On the basis of their number, these cocci bacteria are categorized into different types. What are they? First one is monococci. Only single spherical non-motile bacteria. This one. Diplococcus. This means 2 spherical bacteria are associated. Next one, tetracoccus. Tetra means how many? 4 cocci bacteria are present. Sarcina, that means 8 spherical bacteria. 8 spherical bacteria. And then streptococcus, this means numerous spherical cocci bacteria are arranged in a chain-like structure. Next one, staphylococcus. Number of spherical bacteria are arranged in irregular manner, look like a grapes. Understand? In this way, on the basis of their number of spherical bacteria, cocci bacteria are furtherly divided into monococcus, diplococcus, tetracoccus, sarcina, streptococcus, and staphylococcus bacteria. Rod-shaped bacteria are called bacillus bacteria. These bacillus bacteria are also categorized into monobacillus, diplobacillus, and streptobacillus. Monobacillus, this means only single rod-shaped bacteria is called monobacillus. Diplobacillus means rod-shaped bacteria. Twice 2 rod-shaped bacteria are called diplobacillus. Streptobacillus means member of rod-shaped bacteria are arranged in a chain-like structure. These are called streptobacillus. In this way, on the basis of their number of rod-shaped bacteria, bacillus bacteria are categorized into monobacillus, diplobacillus, and streptobacillus bacteria. Vibrio. Vibrio means this one is a half helical. Complete helic means this one is the complete helic. Half helic. This one is the half helic. Okay. The half helic or comma shaped, comma shaped bacteria are called Vibrio bacteria. Helic bacteria, complete spiral or complete helical bacteria is called spirillum bacteria. Okay, in this way, on the basis of their morphological structure, bacteria are categorized into cocci, bacillus, vibrio, and spirillum bacteria. Understand?